Good. We're all good. Thanks for joining us. We're experimenting today, so I hope that you can hear us. We have some people in person and joining virtually. Welcome to Riot's Lunch and Learn series. My name is Caroline Griffin. I'm Riot's Director of Operations, and I am super excited to have, and I'm just going to make sure that people can, I don't think people can see, can everyone see me?
Yeah.
Okay, you can on there. Okay, okay, perfect. Okay, awesome. I'm super excited to have Torus and GDA with us today. I won't go into explaining about Torus and GDA because the lovely Ashley Valenti will do that for us, but we're so excited to have Ashley and Ken with us today to talk about data and the dirt, the root shape of all things. But I will hand it over to them. If you have any questions, if you're joining us virtually, please do put those in the chat box. Ashley will be monitoring the chat box. And then of course, if you're in person and have questions, please just raise your hand and Ken and Ashley will get those for you. Thanks for being with us, y'all.
Thanks, Caroline.
Absolutely.
All right. Hi, everybody. I'm Ashley Valenti. I am a senior scientist and business development person at Geometric Data Analytics, or GDA, and Torus. So GDA was founded in 2012. We primarily are a government contracting company. And then some of these government contracts started to become SBIRs, which are grants with a more commercial focus. And so Torus was founded in 2018 and is kind of our commercial effort entity. So GDA and Torus are the same group of people at this point in time, but that's why it's both of us. And I also want to mention, so Ken is going to talk about one of those SBIR efforts today, the Root Shape Project. One of the other SBIR efforts, we are enthusiastically currently participating in the Riot Accelerator program, which I can't recommend highly enough if you all are considering that. But without further ado, I'll hand it over to Ken.
Thank you, Ashley. There we go. Great. Yeah, thank you everybody for coming today. My name is Ken Ball. Just a little background about me and then the project, and we'll jump into it. So I'm a NC State grad, actually, so kind of local undergrad and grad school, and I've been at GDA for more than 4 years now. So, I'm a mathematician, and so this project. It started out as a STTR, which is kind of the same as SBIR, except we started out with a Research Institute partner as well. So my research partner on this is Anjali Iyer-Prascusi. She's a plant pathologist at Purdue University. Just a little background, how this came about was browsing RFPs, solicitations, and I remember the DOE, this is the Department of Energy funded project, and I recall browsing through it, and our CEO John Harrer, who's here, he has a textbook on topological data analysis, computational topology, and had an appendix in there where he had done some root analysis. And so I was like, I bet we could do this. And so that's kind of how this all started. Yeah, so I'll jump right into it. So we are now, if you're not familiar with SBIR or STTR process, they are research funding that's set aside by federal agencies that fund research in general. And in addition to addressing a scientific problem that's of interest to the funding agency, there's also a commercialization component baked into the program, which has been really interesting because we're not only thinking about the underlying scientific problem, technical problem, but also how does it turn into a sustainable thing, right? Partly a sustainable business, but also just how is this not something that's written and then lives somewhere on a repository that nobody ever uses, right? So, it's really been a fascinating process. So just a brief overview of what we're going to talk about today. The title of the talk is Root Shape. Well, the project we call Root Shape, and data from dirt, right? So I'll talk a little bit about rhizotrons and the rhizotrons, type of analysis we're doing, the big problem with this, and we'll talk a little bit about the try it at a high level, but I'm happy to address more technical questions as they come up on our approach, computer vision segmentation, to this computer vision segmentation problem, And then a little bit of a few slides just on the product design and how we're actually translating that to our customers' needs. So what is a mini rhizotron? Right, so this is a word we'll see a lot. First of all, I tried to use the simplest possible language for these things. So a rhizotron is an ant farm for roots. Here's an example of such a device from a project that my research partner Angela did. A mini rhizotron is really a transparent tube that you stick in the ground. And the idea is that you plant your plants above the tube, and the roots grow around the tube. So, and then what you can do is put a camera system inside of the plastic tube and take a picture. And the cool thing is you can actually see some of the root growth without digging the plant up. Right? So, yeah, and here is just an example. So this is actually Anjali's set that she's put together for the first phase of our project, where she was growing some tomato plants, and the experiment that she designed was to examine how different tomato breeds responded to some different inoculations, pathogenic inoculations. So, and here are just some examples of the types of images she collects. Right, so this is an earlier experiment from the project with some soybean roots, and this is actually one of the images she collected during our project. And just to give you some idea of what's really going on with these pictures, the top of the tube is actually the horizontal center line of these images. And so just to kind of give you a sense of frankly, these are noisy, difficult images to analyze. We're really going to dig into what that means.
The top of it is on the west side then of that image?
Yeah, like it's almost like if you see my finger kind of going here, this would be like the top. And in fact, on this rightmost one, you see one of the types of artifacts we've seen in images she's collected are condensation bubbles. And so those are pooling kind of at the top of the tube here. And the way that this particular system works is there's the camera system is almost like a scanner that moves side to side or rotationally, I guess, side to side in the tube. And the way that the collection is done is you'll stick the camera into one container, and then you'll just move it further down the tube to the next container, et cetera. And so that way she can do, in this case, she can do multiple containers with different tubes. But the cool thing here is, so these can also be deployed to in-situ environments. So the Department of Energy is funding this project, and pretty much probably to, as far as I know, their biggest experiment right now using mini rhizotron tubes is this spruce experiment. So spruce and peatland responses in the changing environments. This is a experimental setup they have, I think, in Minnesota, actually, with sort of like spruce trees and peat bogs. And they've actually designed these, a number of these chambers where they're kind of have a little ecosystem in each chamber, and they're seeing how it responds to changes in the atmosphere that you might associate with climate change. You have, and so in addition to a number of measurements that they're taking in these environments, they have mini rhizotron tubes that are installed in these kind of around the trees here and also the bog, and they're trying to essentially analyze what happens in the rhizosphere, right, as images are collected. And in these cases, these tubes are probably about 3 meters long and put into the ground at an angle, so generally around 45 degrees. And so they're kind of, again, taking a camera system, putting it in a tube, and collectively and sampling images from each level in the tube. And I'll show later on a sample of like kind of how crazy that image looks when you put it all back together. And this is actually on the bottom of an experiment that's just right down the road at Lake Wheeler. There's some faculty and joint faculty at NC State crop and soil sciences with USDA Agricultural Research Services. And they they're not using it right now, but they have an experimental setup where they have some mini rhizotron tubes in the ground. Similar to this type of experiment. In this case, I think they were growing soybeans or some sort of fruit crop, and trying to see how those plants and their root systems responded to changes in atmospheric warming and ozone levels in the atmosphere. So, and then, of course, as I described before, Anjali is using those container-based systems in her greenhouse. So just kind of a sample of how some people are using these things. The cool thing here is that these are both in situ and non-destructive, right? So non-destructive is really important. Plant roots, I mean, plants are buried underground, like half the plant is underground there, right? Like, and think about how useful and important it is for plant scientists these days to measure phenotypical properties of plants that are growing, like counting leaves, looking at all the stuff above ground. Processes above ground are just as important. And in fact, plants take up nutrients through their root systems. So, but it's largely invisible. And so, you can dig the plant up. There's problems with that. Actually, we'll talk about it a little later. But so many rhizotron tubes and rhizotron systems in general, they're always going to be an incomplete picture because the best you can do is look at the transparent interface of the tubes, where hopefully the roots are growing against that transparent interface. But but it is non-destructive, which means that you can collect time series data and actually observe the dynamics of root rate. And so here is a time series of images that we got from undulating. At the bottom, this is actually a set of time series of images that we did a little bit of alignment on ourselves from Oak Ridge and a little preliminary segmentation on that, trying to align those pieces. But again, this is actually a set of images that is collected over the course of the year in a more natural environment. So the point is, you can also do this in situ. You can actually do things where plants grow rather than just in a controlled laboratory environment. And so here's the big problem. And I'm kind of going through this. Again, please feel free to stop me with any questions. But the really big problem is, how are these things analyzed, right? These are really complicated pictures. And if you really want to do these experiments in an interesting way, I think you need to collect a lot of images. Well, the standard approach is to set your graduate student or lab technician down in front of a computer for about 6 months and have them point and click to annotate these images and say, here, these things are pixels and these things aren't. And there are a few tools out there, software tools out there that can make this a little bit easier and that are designed for this task. But even with those tools, it takes months. And it's also, I'll also add, it's an extremely boring and tedious and terrible task that nobody wants to do. And so not only is it something that you have to engage a grad student or technician or something to do for a very long period of time, but also nobody wants to do. So our solution here, and the gist of the problem, is to automate this process as much as possible. Again, we want to reduce that workload or tedium. We also want you to be able to collect more data, right? So if you're designing your experiment at the outset, you don't want to collect data that's going to take you 3 years to analyze. But you might be able to have more sensitive and more interesting experiments if you can collect more data, right? And I also think that there's a virtuous cycle that we can enable through automation, where the more data that you collect, the better job we can actually do at segmentation itself, segmentation tests itself. So that's part of it. And one thing we've also observed is like being able to do analysis concurrent with the course of your experiment can actually help you identify things that you might need to change or adjust while the experiment is going on. We saw things as simple as in the data collection, whoever was the graduate student or postdoc or tech in our research partner's lab, just had the camera flipped around the wrong way once and got the images out of order, and one image was missing, right? Well, if you wait until a month after your experiment is done to start analyzing your images, then you have a gap in your data, right? But if you have a system where you're uploading and kind of automating that process of analyzing your stuff, you can catch that experiment maybe the day it happens and go back and pull out your data. So there's a number of advantages. Okay, so our technical problem then is automating the analysis of these images. And I would say that it falls under the category of, like the broad category of computer vision. Specifically, we're dealing with the segmentation problem. And these images can be quite high resolution, depending on the camera system that you're dealing with, especially if you want to do things like examine, find root hairs on these root systems, which are specifically things that the Department of Energy is actually interested in. It's also tough to validate results, right? And we'll kind of get into that, but really it results from the complication of the fact that labeled data is expensive to obtain and subjective. And we even observed that in hand annotated images that our research partner provided us with. Our results would differ from those, whatever the graduate student said was a root. But also, if you looked at the raw images, who can say, right? It's not always clear. Like the process of hand annotating these things is actually subjective. And then so we have to validate these results, and then we also have to want to derive meaningful quantitative endpoints for experiments, right? So at the end of the day, the results have to be accurate enough in order to provide the needed sensitivity to the experiments that are being conducted. Yes.
Do you guys have a method for differentiating between root hairs and mycelia?
Not yet, except for the fact that we haven't had images that are high enough resolution to really see the mycelia in the first place.
Oh, okay.
Yeah, that's probably something we're going to be dealing with in phase 2. One of the image collections that we're dealing with, she's going to like bump up the resolution a little more than we were dealing with before. I think there will be an inoculation where we might see some mycelia.
So I'm considering like spectrography to figure out what's actually nutrients and stuff moving through the system and things like that.
I would love to do that. On the software side of things, we're kind of dealing with the images we get, but one thing I would say is it would make a lot of sense to actually do these things with like multispectral systems, and or even just like use the infrared channel and digital camera. There are some groups that are doing that sort of thing on the hardware side. But as in terms of this project, right now, we're mostly focused on the terrible images that we get, essentially.
Hey, you got to work with what you got.
Yeah, exactly. Yes.
Maybe it's outside the scope of sort of building on that. Seems like you might be very interested in looking at what's the other conditions going on. What's the state of the leaves? What's the state of the soil? What's the state of the moisture? The roots may be, you kind of correlate all these other factors, don't you?
I couldn't agree with you more. I think that's a really interesting promise of putting these systems in situ environments, right? So I suspect that they're on the scientific side of things at Oak Ridge in that spruce experiment doing something like that, right? Because they're doing monthly mini rhizotron image collection from what I've seen, and then they also are doing a ton of stuff above ground in those experiments as well. Similarly, our Anjali, in the experiments she did in phase 1, she definitely looked at leaf wilting and some other sorts of things above ground that were happening in the plants to try to infer, especially like whether or not the inoculations, the pathogenic inoculations, took hold the way she expected that they would. But I couldn't agree with you more. I think that the end goal of something like this in the long run would be something that could be like automated in the ground and integrated with other computer vision approaches for.
Well, once you use machine learning for computer vision, that machine learning could accept a lot of additional data as well, right?
Yeah.
It doesn't have to be vision data.
Yeah, absolutely. Yeah. So I think that there's a ton of promise here, and we yes, in the immediate term, we're very focused on just the images that we've gotten, but we have to be thinking about all this because this project ends and we're about 3 months into a 2-year phase 2, and this ends, and we really do want to have something that I think it's fair to say that for our commercialization to be successful, it has to be a bigger vision than the DOE set out for, right? So, but yeah, the underlying complications to the initial sort of segmentation problem here, we'll kind of see some examples that you may have already seen some of these pictures. Soil is unpredictable and heterogeneous, right? And it varies significantly from experiment to experiment, from laboratory conditions to various labs, depending on what field you're in. Yeah.
But I'm curious, even though you're saying it's not, it doesn't affect the living plant, but is there a factor where you got these tubes, does it have an effect on the root growth?
Yes.
So you're kind of you're having some impact on the plants.
You absolutely are. In fact, this is one of the things that both makes them somewhat useful, but also you have to be aware of this. It's almost, the interface between the tube and the soil is actually a low resistance area for the plant roots to grow. So the roots seek out like a path of least resistance to grow. And so they actually have a tendency to grow along the rhizotron soil interface. And so, well, yes and no, right? So it's nice because like you could pretty much be sure that you're going to see some roots, hopefully, if they're growing at all. But I mean, you should take that into account if you're doing this.
If you compare it to previous technology, one of them was you would actually pull the roots up and you would try to x-ray them. You pull a whole structure, yeah, and you try to x-ray, but of course you can't x-ray with water, you get massive noise, so they suck out the water and then it changes. And another one was growing in transparent media and then photographing it. It sounds like a great thing, except that media is represented. I mean, so this is a whole lot better than both of them.
I agree. I think yeah, but it's somewhat. Yeah, until we have transparent soil that behaves exactly the way that something would do out in the field, it's really a hard problem. Yeah, so but yeah, that's actually true. It's somewhat similar to like if you had a big rock. I had one of the people we talked to described it like that. Like it's like, well, I mean, it's not too different from like if there was just like a big rock in the soil and it was growing around that. But it's something to keep in mind. Fortunately, at least it's something you can try to control for, right? So. But you do have to keep that in mind. But yeah, like, you know, as far as like a segmentation task goes, right, we have a really noisy heterogeneous background. It's expensive to obtain labeled data, right? So if you're going to try to do a supervised learning task, it's you know, what good is it to, you know, how much of your underlying data set do you have to label before you can train a classifier on the experiment that you're trying to do, let alone whether or not that's going to be translatable to the next experiment you do. So that's a problem. We also know that there are a number of artifacts in these images that can look like roots. I brought up the example of the condensation, the edges of a condensation bubble, which kind of look like roots actually in that image. But I've heard things from other folks that have done a lot of mini rhizotron collection in real environments, images, and like one thing that was brought up was like termite paths, right? So they're like, oh yeah, we had like, there was one instance where there were like a bunch of termites and you essentially have like dark root-like regions, which are like the termite tunnels like around here, right? So there's all sorts of weird stuff that can happen. Detritus in the soil. A lot of variability over time. One of the things that they're concerned about in some of the DOE, the Oak Ridge experiments that they've done are changing seasonal water table levels. So these tubes are embedded in the ground and go in at a depth, and depending on what time of the year it is, the water table changes, and so that affects your image collection. And then the features are multi-scale. We might be looking for roots depending on the resolution of our image, anywhere from 2 to 3 pixels wide to 100 pixels wide. So there's some similarity across scales, but these are multi-scale features that we're interested in. So just a quick, I literally just copied some images off of Scikit, you know, Scikit images like tutorial on image segmentation. Not because we're going to use this, but I wanted to actually, the real reason I did this is I want to emphasize the difference between our problem and the usual image segmentation problem. And just as an example, though, this is sort of an underlying coin segmentation task. This is just a basic thresholding on the grayscale image, and then this is actually a more like region-based segmentation we do in the tutorial, which uses like a watershed kind of thing that they have implemented in Scikit. The real point I bring up is to differentiate. The reason I bring this up is to differentiate it from our images, because what you have is a relatively uniform background in comparison to the types of things that you're trying to segment out. I mean, well, go back, right? These things are a lot messier than the types of things that you typically see done with segmentation. So, you know, it's not so easy to just use the out-of-the-box segmentation things. Also, and I'll just say our approach to this is actually to try to do most of the work on the unsupervised side. So we're using some differential geometry and topological data analysis tools based around the idea of like what roots look like. what shape roots are, to try to identify the regions of the image that are most likely to be roots. We're also, we actually kind of intentionally at the outset of this tried to not take a supervised deep learning approach. Like, one would think that, and it may be the case, that a convolutional neural network would do okay with this with enough labeled data. Our hypothesis is that just the underlying training of a classifier or essentially a pixel level classifier would be the segmentation task here. With some sort of deep network would require already an amount of labeled data that would be too expensive to obtain. So it really wouldn't be automated. And I don't think it would be transferable from experiment to experiment and from lab to lab, from collection condition to collection condition. We wanted to try to make a tool that everybody could use. So we're taking a unsupervised approach. But then there's also another thing that we recognized as the project was going along, which is that there's only so much you can do with an unsupervised approach. As I mentioned before, there are a number of artifacts that are very root-like, and ultimately we're limited. We're somewhat limited in what we can do without user feedback, right? So I don't know if your experiment is going to have condensation bubbles, right? Maybe you need to tell me that this is a root and this is a condensation bubble. How do we incorporate user feedback in an efficient way that doesn't limit the automated aspect of the process, but rather enhance it? And so on that side of things, we're kind of trying to take an active learning approach where we have a very low dimensional representation of the results of our segmentation task and a process of user feedback where we're essentially filtering out the regions that we think that we think might be roots, allowing the user to train us on that, right, to tell us what they think. So now I have several slides with just images of this thing in operation. This is an ongoing process, so most of these images were actually taken from stuff we were doing last year during the first phase of our project. But I think get across the idea of what we're trying to do. This is a 6-image time series of data that our research partner collected, and you can see what we've done here is we've aligned these images just using some like phase correlation stuff, and then run it through our unsupervised pipeline, and also done a little bit of our own filtering on that with our active learning, where we've taken a very small number of segments that were identified by the unsupervised algorithm and labeled them, and then applied that classifier across the entire data set. And so what you can see is, in this case, root growth that seems to track well with the images that we can parse as units. And a couple more images here. So this is actually, I mentioned the subjectivity of the human classification versus the machine segmentation classification. This was an example of a result that we had on some data that our partner gave us. This was the result of her graduate student's manual tracing. I bring this up to, I don't know how well y'all can see this from the back of the room, but to emphasize the somewhat subjective nature of hand annotation. Are these boundary regions? Are those roots or are they not? Is that part of the root or is it not? Right. So you start to see, especially if you're interested in distributions of root width, you start to see that annotation process, even by a trained technician, which should be the gold standard, is still subjective. It's still going to probably be different from person to person. And then on the right here, if you remember earlier, I had a rhizotron image which was one of the very first slides. So this is the result of that sort of segmentation, where we've done a little analysis on top of that. And you might be interested in root distributions at depth. Right? So these are the types of things we can do very easily once we have segmentations. And I've also, in this case, colored the root segments by their approximate orientation. So you might be interested in lateral root growth versus vertical root growth. So these are all sorts of things we can look at once we've done the basic but very difficult task of segmenting roots out. And then finally, this is actually a stitched together that we did tube from Oak Ridge. Right, so this was on the top. This is the whole image. it's actually about 110, something like that. It's about 110 images that were around 400 by 600 pixels. Each represents a different level that the rhizotron camera system was put into that. So what we did is stitched this together. You can actually, I tried to bring it down in detail so you can kind of see this is the rightmost third, this is like the rightmost ninth of the tube. You can sort of see the stitching is actually a non-trivial problem, and it's important to do because this is something where the systems themselves aren't accurate enough to get everything aligned perfectly. And in this case, these images were only overlapping by hopefully they were overlapping, but by about 5 to 10 pixels. So it was actually a non-trivial stitching process. And then what we have is kind of the result of the segmentation that was applied across the whole thing. But here's some the sort of roots at the very bottom of this rhizotron. And then finally, I just wanted to show what I was talking about with annotation, so getting user feedback. So here's a sample of the image. Here is everything black, blue, and orange is the results of the segmentation. But then the colored things are the results of a user going into our rudimentary application and essentially taking these segments and saying, well, this is a root, this is not a root. So orange is not root, and blue is a root. And so you notice the user said, oh, well, these are condensation bubbles, so I want to label them as such. And there's some little things that are like dirt or pebbles or something like in the root and soil medium, so I want to label those things as orange. And then some of these other things are definitely roots, and I'll label them as blue. So we take a low-dimensional representation of all those segmentation segments, and then train a simple classifier on that, and apply it not only to this image, but to the whole image series. Right. So the cool thing is that not only do we have to we don't have to label everything, and the labeling process is very, very fast. It's can be done in 5 minutes. So the idea is that from the user's perspective, they get these unsupervised results, and they get to fine tune it. They get to filter it. They get to say, well, this is right, this is not right. And then in a very limited fashion, where they only have to do this for a very small percentage of the entire data set. We can learn from that and then apply the classification results that we get across the entire experimental data set. So that's kind of the technical overview. Please, again, if there's any questions, I'll take a sip of my coffee. I'll wrap this up really fast so we can discuss more. But very briefly, how do we take this technical idea to actually being a product? And so, I think first of all, we should ask ourselves, who are our customers? Who are the people that derive value from this? And who are the people that might potentially buy this? So I have a number of customer stories. Some of these were easy because a crop sciences professor, perhaps funded by an industry partner, that's probably our research partner, Anjali. And some of these also kind of came out of our ideas about what a customer might look like for this, that we did our best to validate with the various people that we interviewed as part of our phase 1 process. So I won't read all the way through them, but the idea is that we're trying to identify customers both in an academic setting, government setting, and the industry setting. And we're also trying to think about what are our constraints? What keeps us from getting these things into customers' hands? Rhizotron systems are expensive. The actual, if you're buying it from CID Biosciences or Vienna Scientific or something, these things can be $10,000, $15,000 for the types of ones that they use in laboratory settings. That's probably going to be a barrier for, that's almost certainly going to be a barrier for anybody that's outside of a funded research projects in an academic lab or a government type of lab setting, or a very well-funded industry group that has already has their own scientific research. So maybe are there different? So that makes us have to think about, well, maybe there's some other stuff we can do with this technology, like maybe seaweed, right? So since you brought up, yes.
Or do you see an opportunity where you equipment, it's more like a licensing or leasing situation, and maybe that would be the result.
I don't know. We have, I think we would almost need to identify a use case for that. That kind of happens in academic groups almost, right? Where somebody has a new rhizotron camera system and then somebody else borrows it from them to do their project. We saw that with at Purdue. Yeah, that's a possibility. Yeah.
Were you kind of asking, like, what is the business in this case? Like, how do you guys actually plan to make money with this? Is it like you're going to sell equipment, you're selling software, are you going to sell like software as a service in this case?
Yeah, software as a service. I'll go ahead and answer you. That's our first target. And actually, I think that the tack we've taken on all this, to make a long story short, is that we also want to think about different image modalities that we can analyze with this, right? So if this, I think the first time we thought of this is an interview we had with, a crop and soil sciences professor at State, and they said, well, you're putting the camera into the tube, but what a soil core sample, you take the soil, take a cylinder of soil out of the ground and then analyze it, right? Could you take a picture of that? And so our underlying problem that we're trying to solve is separating roots from noisy heterogeneous backgrounds. Why not even though like the in situ experimental modality is super interesting and like it's very interesting to do this non-destructively, it's also time consuming and expensive. So sometimes it is easier, especially if you're just like out in the cornfield, to pull up a plate. And maybe, what if you can take a picture of the root core and analyze that? Right? What if you have a bunch of soil core samples and you want to analyze that? What if you have scans of roots that you've excavated and you want to analyze those things? Right now, to do that, they have to clean those very meticulously because the scanners and the software that they use on top of the scanners really only work by thresholding, right? And so if you have dirt in there or stuff that's attached to the roots, that just gets assumed. The software that you use just assumes that it's roots. Maybe there's a space for us, and that's it. So actually, that's kind of where I'm thinking we might go, in addition to using the rhizotron systems themselves.
It makes sense that you want to standardize this, like the setup out of the gate, because effectively you have a better chance to, basically teach your computer.
Yes.
And then you want to move to the instance where you're just doing root analysis from any angle, effectively, it sounds like.
Mm-hmm.
And so you see that becoming a machine learning scenario soon, so you can kind of build out your autonomy effectively?
Yeah, I can see it. I mean, so that gets at also an interesting question, I would say, about as we start to develop a customer base, how do people want us to deal with their data, right? Mm-hmm. Are people going to be okay with, we want to protect your privacy. Like if you've done this experimental collection, you might not want us to learn from it, and then, we would love to learn from all of the data, right? But how do we handle that? There's if you're familiar with federated learning ideas, there are some approaches to machine learning where maybe you can try to take something you've learned from these things without really cross-contaminating image sets, essentially. I think that there is I would say that like in the immediate, like this week, we're not really there, but in the long run, I would imagine that as we do more of this for customers in different settings, then there probably is a really interesting supervised or semi-supervised machine learning task on top of all of the image sets. So we pretty much talked about all of these last couple of slides, but actually I do want to bring this up because we did talk about the underlying thing. Our first target here is a software as a service type of product. We want to take that tack because it's easy to support. Like I don't want to deal with, getting some bespoke piece of software to run on on someone's like Windows XP lab machine, right, or whatever. Everybody's different machine, right? I'd rather have something that's like a web app that we're delivering and can work on everybody's computer for everybody's use case, especially because a lot of the computational tasks we have are complicated and can be computationally expensive. Although it's not so bad for us to run it in the cloud for for a given data set that they give us, I don't want our software to depend on very high level hardware for our customer. Right? So, and then we also can be continually engaged with our customers who initially are going to be researchers. And we want to learn from them and how they're using this. But in our interviews, our customers frequently want like a licensed piece of installable software that they can put on their machine and not have to be connected to the internet. In particular, one of the things that came out in our conversations was they're suspicious of systems where they have to upload their image data, not really out of privacy concerns, but because it's usually a pain in the butt. And everybody has their own way of organizing their experimental data. And frequently those things are just they said that their experience is cloud-based image manipulation, image analysis software just isn't reliable enough for them and is too difficult to use. So we're trying to kind of navigate this by delivering a web app that is going to, as much as possible, look like something that's working on their computer. And also we're going to kind of get their data once and but we're not also these are high resolution images. We don't want to be back and forth with them, delivering it back to them every time that they want to pull up the application from the server side to the client side. So in fact, our whole plan of this is actually for the web app to interact with image data that they've stored locally, and so we'll do that by essentially hashing the images. And we know what images that we have our metadata for the images because we've done the processing on it and we have it on our server side. But when they pull it up into the application, we can identify, oh, this is the image you're looking at, and we can deal with image data locally and provide them the metadata of segmentations essentially on top of that. So that's kind of our idea here. But obviously everything's in flux. So I'll just close really quick with announcements or acknowledgements, thank yous, and then take questions. That's okay. This is funded again by the Department of Energy, and thank you, Riot, for having us here. Thank you for having us participate. And I also want to acknowledge our research institute partner, Anjali. And we've really talked to a lot of very generous, very wonderful people in the course of our first and second phase in this project. Just a list of places to get a lot of come from. It's kind of the bottom. So, thank you. Yeah. Yes.
Just a little bit of feedback. I think one thing that you may want to consider is, since you can't really have their information, you are going to be doing the rhizotron analysis on your own servers, and the fact that it's in the back, correct?
Yes.
I feel like one thing that could be contractually dealt with is just the user, I forget what you call it, modified segmentation, where they actually say, that's not a root, that's not a root, that's background image, that kind of stuff.
Yeah.
Right. And that to me presents the possibility of helping other customers, of course, right? Decreasing the time, increasing accuracy, and that's a really easy argument we've made before in previous software and service companies, where we're taking that data and finding the best use case scenarios, right? In our case, we were training kids to stay in university, but optimizing for other customers is usually an easy argument to make. It's your data.
Yeah.
We just want to understand what you don't think is correct, and that's it. We want to be able to take that and to other customers, advancing the field.
Yeah, that's
how semi-supervised learning for that, right? People try to do this in medicine, like, you wouldn't diagnose from these symptoms, when I tell you what you need. Problem is that the doctors are really expensive hourly rates to sit there telling you what to do. So you only want to do this small amount of input from it and learn, include that in your learning. So you can, I think what you're suggesting, it's that knowledge that you get, as you say, here I can conjecture what the ground truth is, and then the verification for users.
Yeah, and I don't think that's a shock to you, but I think that's a critical data point within the business model, right? Necessarily speaking to the long-term IP of what you have, right? Which is going to be this neural net that's very, very educated on what a root looks like, what is a root, maybe even being able to identify additional things in the soil, whatever that may be.
Yeah. I'll take your question. I want to also say, like, to build on that too, we thought about in the sort of client-side software, how do we deal with multiple users within a single lab, right? So you might have multiple people annotating images. You want to be able to share annotations between users, but also keep them distinct. And if I'm the PI on the project, I might be very interested in segments that where different people disagree on what a root is, right? And so that's an additional sort of use case for that. Essentially, like, if we can enable, maybe the point is, well, if you want us to enable that capability, just in our system, that has to be, we have to be willing to share between users, essentially, right? So,
Sure.
Yeah. So, yeah.
Sure.
Yeah. So, yes.
Yeah, great job, by the way. Yeah, with all of these. So, never been such a roots before. So it's really cool.
Yeah.
Sorry to slow before, but if we can like continually take these images or can you set up for frequency, like things that maybe for data, how does that work?
That's a really great question, right? The images that we've seen, right, that we're dealing with so far have literally someone has gone out there with a camera system, and today I'm collecting images. Right. So that's either in the greenhouse or in those field images. In the experiment they did at NC State, they almost had like a video camera that they were just kind of have the video rolling, and that's its own bag of issues, but very interesting image set. But it's the collect right now. Usually, how this is done is the collection is done when somebody goes out there goes to the tubes with the equipment. There's a group that a couple years ago was experimenting with something that I think they called them micro rhizotrons, which were essentially Raspberry Pi camera systems in plexiglass boxes that they buried in I think they were doing it in like a container, but the idea was, well, let's embed this and allow for something that could do continual image collection. I think, and personally, I think the segmentation problem becomes much less complicated the more the better temporal resolution you have. So if we just had a video, like a time lapse, what would almost be a time lapse video of something growing, you could really differentiate soil movement, I think, from roots. Yeah, and also if the things are embedded, it makes the alignment problem a little bit easier, rather than if somebody goes and puts the camera system in every time and they do it a little bit differently every time they do it. So I think that would be a really cool opportunity, but right now, particularly because the experimental equipment that the researchers that do this are using is so expensive, they're really just doing image collection like on the day that they've decided to go out and collect images. Yeah.
Interesting. Yeah. Where do you see this going?
Well, I think I don't want to predict the future, but I think that we have a pretty good grasp on the, we've already seen that our experimental results on the, our quantitative results, even with our very rudimentary pipeline in phase 1, we're able to differentiate the experimental groups that our research partner Anjali did in her experiment, right? So you could say, oh, well, this plant was susceptible to the pathogen, and this group tended to have poor root growth under a number of quantitative measures, right? Generally, like pixel count of root mass. Yes, sorry.
I'm an avid tomato farmer,
Yeah.
citizen scientist. I mean, if you threw this out on Amazon for $100,
yeah,
and I can monitor my tomato roots. I mean, I just think that there's other ways of collecting data, and the citizen science route could be huge for you guys.
Mm-hmm.
And it could be a pretty low
barrier
to access.
I think it'd be cool, even if we didn't want to get into the hardware space, we could at least say, hey, here's like if you're kind of in the maker group community and you've got your Arduino board and your Raspberry Pi and you've got a Raspberry Pi camera, here's how you build a mini rhizotron setup for yourself. And then, hey, if you want to use our software to analyze it, maybe that's the approach to
your website, get the referral fee. Yeah, sure.
Yeah. So
as long as you send your data to us.
Yeah.
I mean, the data is everything. I can't emphasize that enough.
I Couldn't agree more. Yeah, yeah.
Do you think there'd be a point where the data itself could be valuable, or like it's not like you're selling people's personal information, it's information about the roots?
Yeah.
And then what is that business like? Like, are there competing group companies? Are there other solutions competing with what you're doing?
Yeah, I can at least answer that in terms of other people competing, there was another under the same topic, another company did get funded for phase 2. So I know he's one competitor. I think they're based out of Hawaii, if I remember. The right now the landscape of this sort of image analysis software is mostly oriented towards academic people that are in academic settings that are collecting these things, and they're usually it's generally the instrument manufacturers themselves that will have so RootSnap, for instance, so CID Biosciences manufactures one of the many rhizotron systems that we're familiar with, and they have a software product called RootSnap, which they have nice pictures of it. Loading it up on the tablet, you can be on the field and look at what's being collected, but it's still a manual tracing thing, essentially, right? There's some, maybe snap it to a root type of features as you trace out these roots. It tries to do its best, but it's still essentially a manual. You have to manually trace the whole thing in order to use this. There's a company called Regent Instruments or something. They have a number of software products. WinRhizo is their big one, and that's actually one that they oftentimes people use for scanned excavated roots. So but it's essentially it has some annotation stuff built in. People are used to using it, but it's essentially like something that they sell kind of along with. I believe they also make scanners. And so you also can get your license for WinRhizo, which will allow you to essentially it does like a type of thresholding segmentation on the scans because the background for the scan. It's assuming that you've cleaned your root cores essentially. They also have some software called like rhizotron, which is similar to RootSnap, where it's kind of an augmented manual tracing application. In fact, that's what our research partner used for this project to do manual tracing for her soybean roots. I'm sorry, I'm just going through a list of every company that I can think of. There's also there's a couple of open source projects out there. There's one that I think is pretty interesting, but I don't think they're really doing automation still. It's more just like a convenience, like for manual annotation. So there's not really automation out there in terms of the software.
So there is competition, but not automated. Like you're kind of the smart version of it.
Yeah.
Now you said you guys were not yet at least doing machine learning or deep learning.
Not really, except for the sort of user-facing application at the end, where we're taking the low dimensional representation of the segments that have just been labeled as either root or not root.
Okay.
Yeah, but everything else that's done up to that point is unsupervised. There are problems with that we're still working out. There's always a couple of hyperparameters that need to be tuned and stuff like that. But it's besides that hyperparameter tuning, it is unsupervised. So
one of the twists on business Can you compensate for the fact that you can't see it by looking at what it is? So that means collect all this other kinds of data, soil, and you based on that predict. So here, for example, if you're a company plant, typically you grow plants at different stages and you get transport. So if they get root bound back in trees to grow. The level of that happens. If you take the food too early, then you are gone. So, they guess. And they say, what we've been doing for years, we believe this about the right side, and sure this is fine. But are they right about that? People verify that. I'm sure that's completely different. Other things that we do.
do you guys need at this point?
Time. Yeah, well, yeah. I mean, we are, frankly, in terms of where we are on the project, we had a prototype analysis pipeline and a prototype application put together for phase one. And we're in the process of kind of rebuilding that pipeline into like an alpha level software product that's a little more rigorous. So we can kind of do continuous integration and continuous development on that. And then also on the front end side of things, kind of moving from a very basic like dash app that I threw together to like essentially something. Our next step is essentially let's deliver a software product to our research partner, who is essentially our paid customer, right? Let's deliver something to her graduate student and like see how they use it and get feedback. So
Yeah.
And then I'll add from business side of things, I think we want to, validate some desirability assumptions for the academic market specifically, and then not super near term, but look for other markets, like, you mentioned the citizen scientists and things like that.
Yeah.
So just kind of continuing to explore some of the other outside.
Okay.
Yeah, the part of the reason for the academic work is that it's credentialing. So for example, we talk to people that are reading and said, it's the better thing to be interested in. And they said, yeah, it sounds really good, but I wouldn't actually know exactly how to use it before talk. So it's a we say that that's what happens over and over again. So part of the idea is to really demonstrate capability is powerful.
I believe you, and I'm drawing academia for validation too. I've sold in academia too, using ed tech software service, and even serving in like, academia is almost like healthcare, since everybody's buying for money, and there's this other side of it where it's monolithic and moves slower than icebergs.
Yeah.
So academia is an easy way to drive it through the line, unfortunately. So I would say that unless you're ready to build a sales force that has 12-month-long sales cycles, because you're relying on institutional budget cycles, it takes an insane amount of patience. So that's my I've come back to tell you from my war story.
Yeah.
But part two of it is validation doesn't necessarily need to come from that sales cycle.
Yeah.
What other kinds of validation do they look for in terms of your customer targets?
Yeah.
Have you gotten signups? Have you gotten like monetary commitments, either the Kickstarter or even just like small equity portions through like a research council? Like you've been able to get that kind of buy-in early traction that you can sort of show investors, so when it's time to scale, you can present that kind of evidence?
We have not yet. Really, the extent to which we have that sort of evidence would be letters of support that we've collected as part of our letters of support that we've collected as far as our application process for our phase 2 round of funding. But those are they're non-binding, right? So it's
Sure.
It's not quite the same as put your money where your mouth is.
Well, I mean, like a letter of intent saying effectively, like when this comes online, we'll buy this.
Yeah. We do not have that. And I think actually a prerequisite for that is that even though we have a prototype, we don't have yet what I would call our minimum viable product. So I think we have a clear path towards that. And Ashley has been super helpful with defining what that MVP is and getting it out there. But at least in my I would be happy to be corrected on this by other people's experience, but I personally feel like I want to have that MVP to really demonstrate that use case for people beyond just like pictures of our results. Like,
sure. Yeah. I mean, I think there's a good argument to be made that MVPs don't necessarily have to be the actual prototype. You can do smoke tests, obviously, and you can do the video that drops in, and you can see which markets are actually going to commit to this early stage. I mean, it's a beautiful vision, and I feel strongly like I think this would be a very fun business to go after.
Yeah.
It's just, you strike me as someone who's been in academia. Have you spent time in academia?
Adjacent, I mean, graduate school, so.
Oh, so you know about the grad slump. Okay.
Well, so, yeah, so
you're
I'm,
going for your thesis, your dissertation,
Yeah.
and like validation is even hard to come by when you're in it.
yes.
So like all things considered, academia is just tricky. I have done my utmost to steer people towards like earlier validation so they had the investment that buys them the time.
Yeah.
You know, so if you haven't considered like the evidence you're going to present fundraising, and you're going and you're a year out from that, I would start building that as soon as possible because it's going to take you 6 months to get to the point where you're in the room with that investor to present that evidence. Well, I'm just saying you can't be early enough, that's all.
Yeah, no, thank you. Yeah.
That's great.
Yeah, yeah. How are we doing on time, I should ask?
So
Are we-
we're over. I don't
Yeah.
It was really nice to meet you.
Thank you, it was really wonderful to meet you too.
Yeah, thank
Uh,
you so much. Thank you. guys.
Thank you all.
Welcome to City Network.
Yeah.
Please find the time to see that the guys is not your man. Thank you for competing for lunch with amazing. And again, feel free to stay in network.
I'm on the sheet though.
Thanks, everybody.
John Harrow. Sure, I mean,